So good morning students. Now we are just discussing male reproductive system 2. So in this we will discuss the topics left over in volume 1. So first of all in this we will just discuss about the TS of the testes. So in the first volume we just discussed the location of the testis that is present in the scrotal sac outside the abdominal cavity so as to maintain temperature lower than the body temperature, which is essential for maturation and formation of the spermatozoa. Okay. So because testis is the primary sex organ, so certainly it will help in the formation of the sperm. So now just look at the TS of the testis. We know well that testis is made up of large number of seminiferous tubule. So here you can find in the diagram also that this diagram shows large number of here 3 seminiferous tubules are given over here. Okay, so in between these seminiferous tubule you also find the connective tissues. These are the connective tissues which are called as interstitial tissues. It is called a Leydig cell or interstitial tissues. So this interstitial tissue shows the presence of Leydig cell. In the interstitial tissue, we saw the presence of interstitial cell or the Leydig cell, and the Leydig cell are very important because they help in the production of the male androgen, that is the testosterone. The testosterone is essential for the spermatogenesis and also for the development of male sexual character, secondary sexual character. Okay, so now just look the structure inside the that is seminiferous tubule. So inside the seminiferous tubule you can just simply find that it is just being surrounded by epithelial tissue. Okay, so that epithelial tissue, so that's why you just find over the presence of basement membrane. This basement membrane is present outside that epithelial tissue, and this epithelial tissue present inside this seminiferous tubule is cuboidal in nature, and that is called germinal epithelium because it divides to form a spermatogonium or a spermatogonia. The spermatogonia are diploid myocyte cell, means a sperm mother cell. The spermatogonia is a sperm mother cell, and that's why it is diploid in nature. This spermatogonia cell are cuboidal structures, and you also find interspersed between these spermatogonia cell you also find long columnar cell. These are Sertoli cell. These long columnar cells are called as Sertoli cell. They are also called as nurse cell or sustentacular cell. So the question is, how will you differentiate between the spermatogonia cell and Sertoli cell? Sertoli cell are present inside the seminiferous tubule, and this can be differentiated from the spermatogonia cell because Sertoli cells are long columnar cells, clear, elongated cells, but spermatogonia cells are cuboidal spherical cells that both of them arise from the germinal epithelium. But the Sertoli cell in function, it is similar to that of the tapetum that we have already studied in the anther wall. What is the function of tapetum? Tapetum is meant to provide nutrition to the microspore mother cell. In the same way, this Sertoli cell also provides nutrition to the myocyte cell, that is spermatogonium, so that spermatogonia cell may undergo meiosis to produce spermatids, and finally that can differentiate to form a spermatozoa. Clear? So here what we find that inside the seminiferous tubule we can find the different type of cell that has been formed during the formation of the spermatids and spermatozoa from the spermatogonia cell. So what are these cells? That the primary spermatocyte cell, then you find secondary spermatocyte cell, and then you find the spermatids, and even you can find the spermatozoa. These are the spermatozoa. This is the spermatozoa. Okay, so this is the TS of testis, and students please keep in mind that TS of testis is important both from the theory as well as from the practical examination. because in spotting TS of testis is being asked. Okay, so now we are just simply coming to the next topic. The same whatever I have discussed you, the same is given over here. So now just come to the next, that is now the hormonal regulation of the male reproductive system. How the male reproductive system, they are just under control of the hormones. What is the role of different hormones in regulation of the male reproductive system? Okay, so first of all, you know about the hypothalamus. What is this? It is master of master gland, or it is neuroendocrine gland that produces different type of gonad release hormones. It releases different type of release hormone that stimulate the pituitary to further activate other organs. Clear? So that's why during control of the gonads, the hypothalamus produces gonadotropin release hormone. This gonadotropin release hormone, it now stimulates the anterior pituitary. This is anterior pituitary. Now the anterior pituitary under the influence of GnRH releases two type of gonadotropin, LH. LH means luteinizing hormone and FSH means follicle stimulating hormone. Okay, so now come to the luteinizing hormone. Which cells are being affected by the luteinizing hormone? Luteinizing hormone in case of male, it activates Leydig cell or interstitial cell that we have studied just now, that it is present in the connective tissue in between seminiferous tubules. Clear? So these Leydig cell, when they get activated, they releases male androgen called as testosterone. This is the testosterone. Okay, so this Leydig cell releases testosterone, and this testosterone, what is the function? It helps in spermatogenesis, means formation of the spermatozoa, formation and maturation of spermatozoa, as well as it helps in the development of secondary sexual character in case of male. Clear? Now, but when excess amount of testosterone is being produced, then it also negatively affect the hypothalamus. It also negatively affect the pituitary, because it is called feedback control. When excess amount of testosterone is being produced, then it just simply give information to the hypothalamus and the pituitary, so that hypothalamus stop the secretion of GnRH, and if it acts on the pituitary, then pituitary stops synthesis of the LH. Clear? Now just come to the effect of the FSH, follicle stimulating hormone. Follicle stimulating hormone, it acts on Sertoli cell. Sertoli cell, you can recall, long elongated cell that is present inside the seminiferous tubule, clear, which are called as a nurse cell. So this Sertoli cell, when activated by FSH, it produces ABP, that is ABP, androgen binding protein. It produces ABP and also produces inhibin. ABP means androgen binding protein. What is the function of ABP? ABP helps in concentration of testosterone, due to which the spermatids get converted into the spermatozoa. The spermatids feeds on this concentrated testosterone, and because of that it can get converted into the spermatozoa. This process is called as spermiogenesis. Clear? But it can also release inhibin. This is the inhibin, and this inhibin, it just negatively control this system. It results in decreased production of the FSH and also decreases the level of GnRH. Clear? So in that way, this is the hormonal regulation of the male reproductive system, where the role of different hormones you will have to understand. Okay, so now come to the next topic. So next we have to discuss spermatogenesis. Spermatogenesis, also called as spermioteliosis. Spermatogenesis or spermioteliosis. And what is this? Spermatogenesis is the process of formation of a spermatozoa from the germ cell. Germ cell means a spermatogonia cell which is diploid, and it is also called as myocyte cell. And where it occurs? It occurs inside the seminiferous tubules of the testes. But spermatogenesis is completed in two steps. The first step is called as spermatocytogenesis, because in this step spermatocyte cell, primary spermatocyte cell, is produced from the spermatogonia cell. So that's why it is called as spermatocytogenesis, primary spermatocyte and secondary spermatocyte. That we will see in the next slide. And the spermiogenesis means conversion of secondary conversion of spermatid into sperm or spermatozoa. This process is called as the spermiogenesis. Clear? So next I am just discussing you the different steps of that spermatogenesis. Okay. So before coming to the spermatogenesis, I just want to tell you that spermatogenesis is being completed in three different phases. The first phase is called as the multiplicative phase. This multiplicative phase, it occurs by mitosis, and during this what happens that the spermatogonia cell, they divide, they multiply and add more of the spermatogonia cell. But here one point is very essential for you to understand, that the spermatogonia cells are of two types, which is called type A spermatogonia and type B spermatogonia. Type A spermatogonia cell, they just divide and redivide to add more of a spermatogonia cell, but type B spermatogonia cell only takes part in the formation of the spermatid and spermatozoa. Clear? So that is the very first phase, multiplicative phase. It only results in mitosis. It occurs by mitosis, and during this only the number of spermatogonia cell increases. Clear? Now the second phase is called as growth phase. During this growth phase, the size of the spermatogonia cell increases, and now it is called as spermatocyte. Clear? So that is also growth phase. It is accomplished with the help of the mitosis. And now the third phase is called as maturation phase. So maturation phase, it occurs with the help of meiosis, and as you know very well that meiosis is also called as reductional division, that has two different phases. So the first phase is reductional in nature, and the second phase is equational. Okay, so that I will discuss you with the help of diagram. Now just look at the diagram, it will be clear to you. So now here just look at the diagram. The very first phase I told you that it is called as multiplicative phase, during which spermatogonia stem cell, they divide by mitosis so that their number increases. So now just look here, the first diagram, you can just simply look here, that is the spermatogonium cell. They just multiply and they produces two type of that is spermatogonia cell. The type A, they just simply divide and redivide to add more of the spermatogonia cell, and only type B spermatogonia cell, they enter in the growth phase, that is the growth phase. Only type B enters in the growth phase, during which what happens, their cytoplasmic mass increases, and now it is called as primary spermatocyte. After growth phase, it is called as the primary spermatocyte, and it is also diploid in nature because only mitosis has occurred up to the formation of primary spermatocyte. Now this step of conversion of spermatogonia into spermatocyte is also called as spermatocytogenesis, because spermatocyte has been formed over here. Okay, now come to the next step. After growth phase, now we have maturation phase. This is the maturation phase. I have told you earlier that maturation phase includes meiosis. Clear? So that's why the primary spermatocyte now undergoes meiosis. Clear? And after completion of meiosis 1, you know well that meiosis 1 is reductional division. So after completion of meiosis, what happens? Meiosis 1, two secondary spermatocytes are being formed. How many? Two secondary spermatocytes are being formed, and these two are haploid in nature because meiosis 1 is reductional division. So because of the reductional division of primary spermatocyte, two haploid secondary spermatocytes have been formed. Okay. Now the secondary spermatocyte, now they undergo meiosis 2. Meiosis 2 is equational division. So because of that, what happens that each secondary spermatocyte produces two spermatids. Clear? So altogether the two secondary spermatocyte produces 4 spermatids. Clear? So that's why from one spermatogonia cell, after completion of maturation phase, 4 spermatids are being produced. Clear? So one spermatogonia cell will produce 4 spermatid. But look at the diagram here, what this whole step from the that is spermatogonium to the formation of a spermatid, it is the early part of the spermatogenesis. Clear? Where the spermatids have been formed. But the spermatids, as you can see from the diagram, these are non-motile, these are spherical in shape, they are haploid in number, haploid in the ploidy. So because of that, these spermatids now they have to get modified, they have to get differentiated, and after differentiation only they will get converted into the spermatozoa. So now just look at the difference between the spermatids and the spermatozoa. What you find in the diagram, that during this this is called spermiogenesis. During this spermiogenesis, the cytoplasm, here large amount of cytoplasm was present, that has just got reduced. Okay, so here you find that the amount of cytoplasm is less. Second, what you find, there is lot of modification in the mature type of a spermatozoa. You saw the presence of head, you saw the presence of middle piece, this is called middle piece. Then you find the presence of tail. Clear? So these all differentiation occurs, and then only the spermatid get converted into the spermatozoa. So this process is called as spermiogenesis. Clear? So now we will just look at the next slide. In the same, this is more simplified diagram, so I have just simply given to you so that you can just draw the diagram. It will be easier for you. Here I have just simply divided it also in x and y. If you find comfortable, it's okay, otherwise you can just go for the first one only. Okay, so now one more point. Now just look at the enlarged diagram of the seminiferous tubule. Sectional view I have just shown to you. So here just look at the different stages that you have just studied during spermatogenesis. So now just look here, this is the spermatogonium cell, this is the long tubular, this is the long tubular, that is Sertoli cell. Okay, now what you find, that this Sertoli cell, here what happens, I have told you that the Sertoli cell produces ABP, that result in concentration of the testosterone. So because of that this spermatids, just look here, this is the spermatid. This spermatid, it just simply pierces inside the Sertoli cell to just feed on the concentrated testosterone, and after that only it just get converted into this spermatozoa. This is the spermatozoa. So what happens, and after the formation of a spermatozoa, differentiation of a spermatid into spermatozoa, the spermatozoa get detached from the Sertoli cell, and this step of detachment of matured spermatozoa from the Sertoli cell is called as spermiation. We have discussed three different terms: spermatogenesis, that is the formation of spermatozoa from the spermatogonia cell. Clear? The spermatogenesis has late spermatogenesis has one more phase, which is called spermiogenesis, means conversion of a spermatid into a spermatozoa. Clear? And the third term is spermiation, means detachment of matured spermatozoa from the Sertoli cell is called as spermiation. Okay, so now just look at the next slide. The same definition I have given over here. Spermiogenesis is the process of transforming a spermatid into mature spermatozoa or sperm, and spermiation means sperms get attached to Sertoli cell to draw nourishment and finally get released from the seminiferous tubule by the process called as spermiation. Okay. So now just look at I am just showing you the changes during the spermiogenesis. Actually what happens, just look at the first diagram where you find that this is a spermatid which is spherical structure, clear, and this is the matured one where you find the presence of head, presence of middle piece, presence of tail. So here how this modification has occurred. So here what happens, there is a great role of the Golgi body. So Golgi body, it just simply results in formation of acrosomal vesicle. It is given over here, acrosomal vesicle, which is present at the head of the that is sperm. Okay, so here what happens, that the Golgi body help in the formation of acrosome of the sperm, which is present at the head of the sperm. And then what happens, centriole are also present over here, that centriole are present in the pair and that help in the formation of the axial filament, and because of that finally the tail is being formed. Okay, in the middle piece concentration of the mitochondria is found. So this mitochondria, they help in providing energy so that the sperm can just show motility. Okay, so these are the different changes. You can just simply find how the amount of cytoplasm has got reduced and the spherical spermatid got converted into the long elongated sperm. Okay, so now just look at the different parts of the sperm. So here what we find, that the sperm has different parts. The very first part is called as the head. This whole part from here up to this, it is called as the head. So head shows the presence of nucleus. It has small amount of cytoplasmic mass in which nucleus is present, and nucleus contains haploid set of chromosome, and this nucleus only take part in the fertilization. It can just simply fuse with the female pronuclei and result in formation of gamete. Okay, now at the tip of this head, at the tip of this nucleus, you find at the tip of the head you find the presence of one cap like structure. This is the cap like structure which is called as acrosome. This acrosome is very important because the acrosome shows the presence of a different type of hydrolytic enzyme collectively called as sperm lysine. Clear? That contain the enzymes like hyaluronidase, Jonah penetrating enzyme, Jonah lysine. So these all enzymes are hydrolytic enzyme which are meant to penetrate and degrade the walls present around the egg. So that we will discuss while discussing about the female reproductive system. So because at that time it is important because acrosome contains different type of the hydrolytic enzyme collectively called as sperm lysine, and these enzymes are being produced by the Golgi body. Okay, so now next to that you just simply find the presence of next structure. You can just simply find this is the middle piece. This portion is the middle piece. Middle piece shows the concentration of large number of mitochondria. Large number of mitochondria are concentrated in the middle piece so that the mitochondria can provide energy for the sperm to swim. But between the head and the middle piece, one very small structure called as a neck. Neck is very important because neck shows the presence of centriole. Centriole, as you know, that always present in pair. So that's why in the neck region here two centriole is present. That is proximal and the distal centriole. And these centriole are very important because the proximal centriole is very important because one thing you will have to keep in mind, that the egg produced by the female reproductive system or the ovary do not contain any centriole. Okay, so that's why what happens for the division of the zygote, the centriole, proximal centriole which is present in the neck of the sperm, it is important because it can only help in the division of the zygote. Clear? And the distal centriole of this spermatozoa, it help in the formation of the axial filament that finally result in formation of the tail. Clear? And tail is very important because it help in it provides the motility so that the sperm can swim. Clear? So this is all about the structure of the sperm. So by this we have finished the male reproductive system. Okay, so after that I'll give you some of the assignment. If you got any doubt, you can ask the questions. Okay, so thank you.